All right, welcome to Chem 313, Lecture 12. To review last lecture, we talked about cell signaling or signal transduction. And what I said that you're responsible for knowing from that part of the course is to understand what are the common features of all cell signaling processes. That's what I'm going to test you on. So what are these common features? First of all, we need to have a signal, which is just some sign of a change in the environment or in some sort of conditions that the organism is living through. Second is a receptor, which is just something that picks up the signal. So if your signal is a molecule, such as a hormone, then the receptor usually binds to the signal molecule. Third thing is a relay, which is some sort of process for the signal going from the outside to the inside of a cell. We have target activation, which is when proteins that carry out the desired function get activated. Or in some cases, inhibited. And then lastly, we have termination, which is when the signal stops. And that one's pretty self-explanatory. So, as an example, last time we talked about the epinephrine signaling pathway. And one of the outcomes of epinephrine signaling was the release of glucose from glycogen, which is a molecule that is used to store glucose. And why would that be an outcome of epinephrine signaling? Well, remember that epinephrine signaling occurs during acute stress. In acute stress, you need to be able to run or fight or something like that. So we need our muscles to do work. And your muscles doing work requires fuel. And glucose is an important fuel molecule. So that's why release of glucose from glycogen is an outcome of epinephrine signaling. Today's lecture deals with carbohydrates. And we're going to talk about all of carbohydrates in a single lecture. I'll be honest with you, I find carbohydrates to be incredibly boring, so I'm going to go very quickly through this subject. We're going to talk about very briefly the structure of carbohydrates, at least of the simplest carbohydrates, which we call the simple sugars or the monosaccharides. We're going to talk about the very basic chemistry of carbohydrates, but only in extreme basics, and its purposes in living cells. And there are two primary purposes of carbohydrates. One is as a fuel source, in other words, food. And the second use of carbohydrates is structural. So in some organisms, that means cell walls. So in plants, algae, that sort of thing, cell walls are made of carbohydrates. In animals, we don't have cell walls, but we have extracellular space. So the extracellular fluids of animals are surprisingly carbohydrate rich. All right, so for this topic, I would rather outsource this to a worksheet than do a lecture on it, because the lecture is really not going to cover anything you couldn't learn in a worksheet. So I have a carbohydrate worksheet, which is up on Canvas. I borrowed it from somebody else, so I don't have a key. But the carbohydrate worksheet, if you can get through that, you should be in good shape for any carbohydrate question I will ask you on any quiz or exam in this class. Still, the goals for the carbohydrate section, things that you should be able to do by the time we finish with this unit. You should be able to distinguish ketone sugars from aldehyde sugars. And that's harder than you might think at first glance. Know some terminology. In carbohydrate chemistry, almost all boils down to terminology. So you should know the meanings of the terms anomer, epimer, diastereomer, D and L configurations, and so forth. You should be able to identify alpha or beta anomers. We're going to talk about that, what that means later. You should be able to identify glycosidic linkages. What that is, once again, we'll talk about later. And also reducing versus non-reducing sugars, which is related to glycosidic linkages. And then lastly, you should understand in general terms the physical properties of polysaccharides. And the 4 polysaccharide classes that I care that you know about are starch, glycogen, and those two are almost the same thing, glycosaminoglycans, or GAGs, and cellulose. All right, so let's take a look into the exciting world of carbohydrates, and that was sarcasm. All right, so what are carbohydrates? Carbohydrates are polyhydroxy ketone aldehydes that have an approximate formula of CH2O n times. So that fact that most carbohydrates have an approximately CH2O n formula led some to believe once that they were hydrates of carbon, which is now not known to not be true, but that's where the term comes from. But they also sometimes will contain other elements like nitrogen, sulfur, or phosphorus. Here's what you should think of about carbohydrates. So the simplest carbohydrates are the so-called monosaccharides, or also known as simple sugars. And a monosaccharide is a chain of usually 4 to 7 carbon atoms with 1 carbonyl, almost always on carbon 2 or carbon 1, and OH groups on all the other carbons. Those are the simplest sugars, and we call those the monosaccharides. So, for example, this molecule would be a monosaccharide, right? We've got 1, 2, actually the numbering starts from this end, 1, 2, 3, 4 carbons. One of them has a carbonyl on it, that's an aldehyde in this case, and the other 3 carbons all have OHs on them. So we have a special way that we would like to draw this carbohydrate molecule to make it a bit more obvious what its shape and configuration is. And that is we draw it as a chain with the carbonyl group at the top, or as close to the top as you can get it. So we would draw it like this. We have an aldehyde So that's the first carbon. There's our next carbon, last carbon. Okay, so what are on each of these carbons? Well, we like to draw them in this format that we call Fisher projections. And the Fisher projection is what the molecule looks like if you were to stretch out the chain of carbons and get it so that everything branching off of the carbons stuck out of the page at you. So this is the so-called Fisher projection structure of this sugar drawn up here. And we have kind of a shorthand way of drawing this. We'll write, since the top is carbon, hydrogen, oxygen, CHO, we will sometimes write it as just CHO, CH2OH, HOH, with the understanding that these bonds on the sides are coming out of the page at you. So that's how we would draw the Fisher projection structure of a simple sugar. We have one carbonyl, only one, you never have more than one in a naturally occurring sugar, and you have OHs on all the other carbons. So that's a monosaccharide, or a simple sugar. So in this particular case, the carbohydrate that I drew is an aldehyde. And there's a special term for aldehyde sugars, and that is aldose. If we had drawn a ketone, sugar, the term for a ketone sugar is a ketose. So what does the Fisher projection structure for a ketone sugar or ketose look like? It would look something like this. Remember, a ketone has the carbonyl group with carbons on both sides of it. So this would be what the Fisher projection of a ketose would look like. Something like that. So what this diagram is showing at the bottom is examples of different carbohydrates. So we've got 3 carbon carbohydrates, which would be known as trioses. In the middle, we have 6 carbon carbohydrates, which are called hexoses. And on the right, we have 5 carbon carbohydrates, which are called pentoses. So if you know your Greek prefix for number, it will help you in this part of the course. So notice that each sugar has an awful lot of chiral centers. And there's one chiral center in particular that we care about, and the reason we care about it is that in every sugar you're going to see, it's always the same. And that is the last chiral center from the carbonyl group. So this one is special in a certain way. So what's special about this chiral center? So the last chiral center, so that farthest away from the carbonyl, it's called the DL center. Why is it called D or L? Well, it's by comparison to glyceraldehyde, which is a molecule that also has a chiral center. One form is called the D form, one form is called the L form. In most, not all, but most natural sugars, if you draw the Fisher projection in the proper way, which is the carbonyl on top, the OH is going to be on the right. And in sugars in which the OH is on the right, that is said to be the D configuration. So you'll recall that when we were talking about amino acids are chiral, and all naturally occurring amino acids have the L configuration, at least those that are found in proteins. For carbohydrates, it's the D configuration that you almost always see. And it always means that if you draw your Fisher projection properly, which is with the carbonyl group at the top or near the top, then the last chiral center, if it has the OH group on the right, that means it is a D carbohydrate. And in fact, almost all naturally occurring sugars on this planet have the D configuration. All right, so let's get into a little bit of terminology. So carbohydrate chemistry is 95% terminology and 5% other Bordeaux. So we're going to just get the most important terms out on the page right now. It's possible, we already saw the distinction between aldoses and ketoses, right? An aldose is an aldehyde, meaning that it has that functional group in it. A ketose is a ketone, meaning that it has that kind of a group in it. We already saw before hexose, heptose, etc., which is naming sugars by the number of carbons. So for example, pentose, we said, has 5 carbons. And it turns out that you can combine these two to come up with new words. So for example, an aldohexose is a 6-carbon aldehyde. So glucose, which is the most common sugar of them all, that's an aldohexose, a 6-carbon aldehyde. A ketopentose would be what? Ketopentose would be a 5-carbon ketone, and so forth. All right, we already saw the D versus L configuration. This specifies the configuration on the chiral carbon farthest from the carbonyl. And that introduces some other issues of stereochemistry. So we have this special term that we call epimers. Epimers are sugars that differ in configuration at one carbon atom, only one carbon atom. And that one carbon atom cannot be the DL center, and it's not the carbonyl, because the carbonyl is not chiral. All right, so sugars that differ in configuration at exactly one carbon atom and are the same at every other carbon atom are said to be epimers. So for example, glucose versus galactose. They differ in configuration at one carbon atom, not the DL center, not the carbonyl. Any sugars that differ more than that are said to be diastereomers. And the term means a slightly different thing in carbohydrate chemistry as opposed to general organic chemistry. So these are sugars that differ in configuration at 2 or more carbon atoms. All right. So those are diastereomers. So how would we compare sugars to see if they're epimers or diastereomers? you would have to look at the Fischer projection structures. So, for example, let's draw the Fischer projections of a couple of different sugars. So here's one sugar. All right, quick, what type of sugar is this? Aldose, pentose, ketose, what is it? This one is an aldopentose. This is actually D-ribose, which is one of the most important sugars out there. And here's another one. I don't remember what this sugar is called. All right, so are these epimers, diastereomers, something else? The answer is these are epimers because they differ in configuration at one carbon atom, and that would be this one right here. Notice that they both have the same configuration at the DL center, so they are both D carbohydrates. So these two are epimers. Here's another example. All right, so this is an aldohexose in this case. All right, what are these? Epimers, diastereomers, something else, same sugar with DL configuration? Well, the answer is these are something else, right? By the organic chemistry definition, these would be diastereomers, but the truth is this one is an aldose, and this one is a ketose. And by the rules of carbohydrate chemistry, they only count as epimers or diastereomers if they're the same type of sugar. So these are completely different carbohydrate molecules. However, you'll note that they have exactly the same empirical formula. Both of these would be C6H12O6. So obviously, it's very difficult to tell what type of sugar you have just based on its chemical formula. You have to look at the structure in the Fischer projection form. All right, so Fischer projections are great because they make it obvious at a glance whether you have an aldehyde or a ketone, or whether you have two different anomers or whatever. But there's a problem with Fischer projections, and that is very few sugars exist in a linear form, at least in water. Most sugars are cyclic. Why are most sugars cyclic? Well, the reason for that is because hydroxyl groups and carbonyls can undergo a reversible reaction to give us something that looks like this. All right, this group is what's called either a hemiacetal, which is the case when R is a hydrogen, or a hemiketal, which is the case where R is a carbon. So you'll notice that carbohydrates by definition contain both carbonyls and hydroxyls. So this can happen intramolecularly. We get an intramolecular reaction, which leads to reversible formation of a cyclic sugar molecule. So for example, I'm going to draw our Fischer projection this way. if this is our Fisher projection for our carbohydrate, what can happen is any of these potentially hydroxyl groups, let's say for the sake of argument that this one will come up and attack nucleophilically this carbonyl group, and the result will be a cyclic sugar molecule. And if that happened for this particular sugar, it would look something like this. So this structure that I've drawn here is something that's called a Haworth projection. And Haworth projections are usually how we draw sugar molecules in practice. And that's for the cyclic structures. And most sugars are going to be cyclic under most circumstances. So the thing to notice about a Haworth projection is it's not supposed to be viewed from the top. What's going on here is that you can think of the Haworth projection as sort of lying at a diagonal or at a slant, where the front carbon-carbon bond is sticking out of the page at you, and the back is pointing into the page. So it's like you're looking at this thing sort of edge on, so to speak, so that these groups in the front are pointing towards you and the one in the back is pointing back into the page. And you'll notice that if you draw a cyclic structure like this, you end up with some substituents, some of the H's and OH's below the plane of the ring and some above the plane of the ring. All right? So that's what a Haworth projection is. Now, Haworth projections are realistic. They're not perfect, but they're realistic. But you'll notice that there's a problem with the Haworth projection, and that is it can be hard to tell, at least without practice, if the sugar is a ketone or an aldehyde. Now, I can tell you just from experience that this one is an aldehyde. Okay, how do I know that? Well, I knew that because I know how to draw Haworth projections starting from Fisher projections. So I do not expect you, by the way, to be able to draw Haworth projections starting from Fisher projections. So that's not something that you will be tested on. However, there is one important thing about Haworth projections that you do need to be able to do. And that is you need to be able to spot this carbon atom right there that I labeled with the star. That's a special carbon atom. That atom is what we call the anomeric carbon. What's special about the anomeric carbon? That's the carbon that used to be the carbonyl carbon. And the anomeric carbon is important because the anomeric carbon has two possible configurations for a given sugar. All right, two possible configurations for a given sugar. So let's talk about that for a second. So normally, you will see examples of this where I don't do this in this lecture. Usually, Haworth projections are drawn with the anomeric carbon on the right and the ring oxygen in the back, right? Because every sugar molecule, when it's in a cyclic form, is going to have one oxygen in its ring. So if it's drawn this way, okay, you can distinguish your two possible configurations of the anomeric carbon this way. If the anomeric carbon has its OH pointing down, we have what's called the alpha configuration. And if you have the OH pointing up, we have what's called the beta configuration. And these two configurations are known as anomers. So what we've drawn down here is the alpha anomer of this particular sugar because this OH is pointing down. The anomers can interconvert. It's not like epimers. Epimers are always epimers. They're never going to interconvert into one another. Anomers can interconvert, and the reason for that is because this cyclization process is reversible. The molecule can open back up and then reclose, and when it recloses, it may reclose with the OH pointing in the opposite direction. So anomers can interconvert. They are considered different configurations of the same compound, but they're difficult to isolate from one another. Okay, so that's great, except the question you may be asking yourself now is, well, wait a minute, how do I know which carbon atom is the anomeric carbon atom? Easy. Anomeric carbon atom is easy to spot. It's always the only carbon atom that is bonded to two oxygens. Look at the whole molecule. Every single carbon atom in there, except for this one, is bonded to one oxygen at most. The anomeric carbon is the only one that's bonded to two oxygens. Now, usually we draw Haworth projections with the anomeric carbon on the right, but we don't always do that. So if you want to be safe and figure out which carbon atom is the anomeric carbon, you have to look for the one that's bonded to two oxygens. That's the one that used to be the carbonyl, and that's the one that can exist in one of two configurations. All right, so this slide shows you a bit more detail on what I just described on the last slide. We have this intramolecular cyclization of sugars. So this process always results in either a 5-membered ring or a 6-membered ring, because those are especially stable for carbon. So if you get a 5-membered ring, it's always going to be 4 carbons in the ring and 1 oxygen in the ring. And when you form a 5-membered ring of this sort, that type of ring is referred to as a furanose, by analogy to furan, the cyclic ether. So, for example, D-fructose likes to cyclize to form D-fructofuranose, which has a 5-membered ring. Okay, we've got 1, 2, 3, 4 carbons and 1 oxygen in the back. A 6-membered ring is always going to be 5 carbons plus 1 oxygen, and that's referred to as a pyranose. Now, an important thing to note is that most sugars can form either a pyranose or a furanose structure. So usually one is preferred because of steric constraints. But it's typically possible to form more than one structure. And remember, for each one of these, you have both an alpha anomer and a beta anomer. This would be the alpha anomer, and this would be the beta anomer. Right? We know this is the alpha anomer because that's our anomeric carbon right there, and the OH points down. This is the beta anomer because our anomeric carbon is there and the OH is pointing up. But fructose could also cyclize to give us 6-membered rings or pyranoses, and in that case, we also have 2 possible configurations, the alpha D-fructopyranose and the beta D-fructopyranose. All right? So we go from one possible sugar molecule to 4 possible rings. And that means that we've introduced quite a bit of structural diversity without ever changing our chemistry in any sort of way. So once again, from one sugar, we can form 2 possible rings and 2 possible anomers. The anomers and the rings can all interconvert, so all of these should be considered different forms of the same sugar. All right. Now, once again, this process, I'm not going to teach you guys how to do this. You do not have to draw Hayworth projection structures starting from Fisher projection structures. That's not going to be done. However, you should be able to identify anomers at the very least. You should be able to tell me whether you have a furanose versus pyranose. And you should be able to understand why all of these different sugars can interconvert as we've drawn them here. All right, so far all we've done is talk about monosaccharides, but let's talk about multimers of saccharides. So a disaccharide is a sugar molecule composed of 2 monosaccharides. An oligosaccharide, oligo means few, so that means that we've got, 2 to, we'll say 10 monosaccharides. But they're all together into a single molecule, which means we need some way to link all these things together. And the way that we link all these together is through what are known as glycosidic linkages. So what's a glycosidic linkage? It's easier to draw an example than it is to explain it. Here's what a glycosidic linkage looks like in practice. Let's draw 2 sugars, and for the sake of argument, we'll say that they are both pyranoses, although they don't have to be. I'm only going to draw one of the OH groups in order to save time. Okay, so here's our first sugar. Here is our next sugar. Let's say that this one has an H and an OH right there. So what happens in a glycosidic linkage is the following. The OH group on the anomeric carbon of one sugar essentially plugs right into the OH group of another sugar. So we have the OH from the anomeric carbon of 1 to any OH group on the next sugar. And these react, they form a structure that looks something like this. So that essentially our 2 OH groups combine into a single oxygen atom, so it looks kind of like an ether, and water is released as a byproduct. This is now a glycosidic linkage. All right, so glycosidic linkages always involve at least one anomeric carbon. And the reason for that has to do with the mechanism of this reaction, which we're not going to talk about. All right? But now we have a glycosidic linkage down here. So you can see a glycosidic linkage looks an awful lot like an ether. It's actually not an ether. It's either an acetal or a ketal, if you remember your organic chemistry. but we're not going to talk about that in any sort of depth. So let's take a look at an example sugar. Let's take a look at lactose. So where's the glycosidic linkage in this sugar molecule? Well, it's right there, okay? This is one of our anomeric carbons, and in this case, the OH has plugged into a different carbon atom on the other sugar. Now, you could have a case where one anomeric carbon plugs into the other anomeric carbon. That's perfectly allowable, but that's not what's happening in this particular case. Now, there is a system for naming glycosidic linkages. Glycosidic linkages always have a name that is something like, for example, alpha 1 to 6, something like that. So notice that we always either have an alpha or a beta to begin with, a number, which is always going to be 1 or 2, an arrow, and then another number at the end. So what do each of these refer to? Alpha or beta refers to which anomer that you have on the carbon atom that is the anomeric carbon, because remember, glycosidic linkages always involve one anomeric carbon. 1 or 2 is the number of the anomeric carbon. Then we have an arrow, and then 6 is the number of the carbon that you are linking to. All right, so why is the anomeric carbon always 1 or 2? Well, that's because if you go back to your organic chemistry textbook, if you are assigning numbers to carbons, the carbonyl carbon gets the highest priority. And in all naturally occurring sugars, the carbonyl is either on carbon 1 if it's an aldehyde, or carbon 2 if it's a ketone. So that first number is always going to be either 1 or 2. So let's assign numbers to this glycosidic linkage, well, to all the carbons in both of these sugars. Why not? So we have our anomeric carbon, which we start off with, and the anomeric carbon is carbon 1. All right, how do we know that it's carbon 1? Well, it's always either 1 or 2, and we see that it's only bonded to one other carbon, so therefore it must be carbon 1. That means this is carbon 2, 3, 4, 5, and then 6. Notice you can have carbons outside the ring. Let's go to the other sugar. This would be our anomeric carbon because it's bonded to 2 oxygens. So that's 1, 2, 3, 4, 5, 6. All right, and what anomer do we have? Well, the OH group is pointing up, so that means we have the beta anomer. And so that means that the glycosidic linkage that we have in this molecule over here is beta 1 to 4. All right, so these are galactose and glucose respectively. And if you number your carbon atoms like I already did, then we discover that we have 2 monosaccharides linked by a beta 1 to 4 glycosidic linkage. So the generic term for this sugar would be galactose beta 1 to 4 glucose. You are not responsible for identifying monosaccharides just based on their structures. That's crazy. Luckily for you, this sugar has a common name, and the common name is lactose. This is the milk sugar. All right, let's have another example of how to identify glycosidic linkages. All right, this is maltose, another disaccharide. Notice, by the way, the wiggly line down here. Sometimes you'll see that. The wiggly line means that you've got either alpha or beta. We don't know. We don't care because in this case they can interconvert. Okay, so that's what that wiggly line means if you're wondering. So let's have a look at it. What type of glycosidic linkage do we have here? Well, I think the best bet is to start off by numbering your carbons. 1, 2, 3, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. So notice it's going from 1 to 4. And in the case of our anomeric carbon, the OH is pointing down, so it must be alpha. I will say that this is an alpha 1,4 glycosidic linkage. And that's what it is. So maltose is properly known as glucose, alpha 1,4 glucose. If you want to do some practice problems, see the carbohydrate worksheet. All right, so this issue of glycosidic linkages leads into this other sort of topic, and that's the topic of what's called reducing and non-reducing sugars or polysaccharides. So this is something that comes up in the literature a lot. It's an awfully outdated concept, but it somehow persists in textbooks, and it comes up in some contexts, especially like in food science, so I think it's worth talking about, at least in passing. So it turns out that carbonyl-containing compounds can reduce copper. Specifically, they can reduce copper 2 ions. So the way it works is something like this. What ends up happening is our carbon-containing carbonyl compound gets reduced to a carboxylic acid, gets oxidized, to a carboxylic acid, and the copper gets reduced. And if this is done in aqueous solution, copper 1 compounds are not very soluble, so they tend to form a precipitate that settles out of solution. All right. So notice that carbohydrates, simple sugars, are carbonyl compounds, and some of them can reduce copper like this. Okay? The key is you actually have to have a carbonyl group, that is, a carbon double bonded to oxygen. If you have that present, your sugar can reduce copper 2. That is said to be a reducing sugar. All right, so which sugars have the carbonyl in them? Well, if you're thinking they all do, it's just hidden, you're on the right track, right? You'll recall that formation of these cyclic structures is reversible. We can always end up with our open-chained form of the molecule, which can then reconvert back to a cyclic structure, right? And because we have a carbonyl group in our aldehyde in this case, this molecule can reduce copper. Therefore, it's reducing. Okay, so I will tell you right now that all monosaccharides, all simple sugars, are reducing. Disaccharides and polysaccharides may or may not be reducing. And the reason for that is because this reaction is reversible only if this carbon is not in a glycosidic linkage. If it is, then it is locked permanently as a ring. It never opens back up to give you the open chain form that has the carbon double bonded to oxygen in it. Okay? So any anomeric carbon that is in a glycosidic linkage cannot reduce copper. All right, so a non-reducing sugar, therefore, is any sugar is just the general term for oligosaccharide, in which all anomeric carbons are in glycosidic linkages. Okay, how do you tell if it's in a glycosidic linkage? Okay, if the anomeric carbon has an OH, it's not in a glycosidic linkage. All right, so in other words, the way that you would spot a reducing oligosaccharide is it is something that has an OH on at least one anomeric carbon. It's non-reducing if no anomeric carbon has an OH. It's that simple. So over at the right on this slide, we have 3 separate disaccharides. I'm going to have you guys find the non-reducing sugar in this list of 3. I'm not going to give you the answer, but if you want to check with me afterwards in office hours or something like that, you're welcome to do that. So which one of these 3 is non-reducing? The other 2 are reducing, and you should be able to explain how you can tell. All right, so the business of reducing and non-reducing sugars becomes somewhat important when you end up with polysaccharide molecules like this one, where you've got a big long chain of carbohydrate molecules. Now, typically, when you have carbohydrate molecules linked together in a chain like this, there's going to be one end of the molecule that has an OH on its anomeric carbon, and therefore that end of the molecule is said to be the reducing end of the polysaccharide. The other end does not have OH on its anomeric carbon, right? There's the anomeric carbon, and you'll notice that there's no OH on it. So this is said to be the non-reducing end. And there are some cases where you have branched carbohydrates. We'll see those on the next slide. And in the cases of branched carbohydrates, typically there is only one reducing end, but there are multiple non-reducing ends. And that has to do with the chemistry of how these complex molecules are put together. We're not going to talk about it. All right, so why do we care so much about the glycosidic linkages and naming the glycosidic linkages and all like that? Well, the linkage, the glycosidic linkage, determines the geometry of the molecule. And the geometry of the molecule determines the conformation of the molecule. And the conformation of a molecule, just like with proteins, determines its physical properties. And so now that we've gotten the basic chemistry out of the way, we can talk about the conformation and the physical properties of polysaccharides. So polysaccharides mean multiple monosaccharides. And based on the conformation of these things, they can have various functions. So we have storage polysaccharides. Storage polysaccharides are typically water soluble, and they exist in a helical conformation. And so these are simply molecules that are used to stuff glucose into. And the 2 important ones of storage polysaccharides are glycogen and starch. And then there's structural polysaccharides. So structural polysaccharides are either rigid and tough, or they are gelatinous in water. because of their conformation, and therefore they're used for structure, especially for cell walls. But insect exoskeletons, for example, those are made partially of carbohydrates. And we also said the extracellular space is filled with gelatinous structural polysaccharides. And these difference in properties all come down to the difference in the shape, which has to do with the shape of the glycosidic linkages. So let's take a look at the storage polysaccharides first. Storage polysaccharides, they're all quite similar. So starch is the structure that I'm showing you here. Glycogen is just like starch, but more highly branched. So what is starch? So starch is a polymer of glucose with alpha 1 to 4 linkages. All right, and it occasionally has alpha 1 to 6 branches attached to it, but for the most part, most of your glycosidic linkages are alpha 1 to 4. So you'll recall that the alpha glycosidic linkage has the OH pointing down, so that when you link two sugar molecules together, you'll end up with something that looks like this. Right? Now, this is an example of why Haworth projections are not perfect. So the rings in Haworth projections are not flat. They are actually, much like the ring of cyclohexane, they're actually crimped a little bit. So because of this alpha 1 to 4 glycosidic linkage, whenever you have an alpha glycosidic linkage in glucose, it may not be the same in all other sugars, the glycosidic bond is going to be axial, right? You remember your chair conformations of 6-membered rings. At each carbon, the substituents, one of them is said to be equatorial because it's roughly lying in the plane of the ring, and the other one is said to be axial because it's lying above or below the plane of the ring. And you can keep going around the ring and draw in the rest of them if you wanted to do that. So when you have an alpha glycosidic linkage, if you're dealing with glucose, the glycosidic bond is axial. So as a result of that, every time you have a glycosidic bond, the molecule gets bent. And so what does that mean? Well, that means if you extend this whole thing into a big long polymer, at each sugar you introduce a bend, so the molecule has this big, long, open helical conformation like so, with some branches coming off the sides. Something like that. So don't think of this as being like an alpha helix for protein. Alpha helices are very, very tightly coiled, and you have hydrogen bonding between amino acids. We don't have hydrogen bonding here with anything except water. It's very open in conformation. It's open enough, in fact, that iodine molecules can bind inside the helix. And so you may have seen in some classes the starch test for identifying the presence of starch in a sample is you take a slice of a tissue or something and you drop a little bit of iodine on it, and it changes this deep dark color. That deep color is because of the iodine binding inside of the starch helices. So because it's open and water soluble like this, we have a kind of spongy molecule that is used to store glucose. And the function of these molecules, both glucose, or both starch and glycogen, is that the glucoses can be cut off the ends and used as a fuel source, like we saw in epinephrine signaling, in which glucose is released from the end of glycogen, one sugar at a time, so that the cell has sugar it can use as food. All right, structural polysaccharides. These are either rigid, and the example of rigid and tough polysaccharides would be cellulose and to a lesser extent chitin, or they can be gelatinous, and we'll see those on the next slide. So let's talk about the rigid polysaccharides first. The most important of these is cellulose. So cellulose is poly beta 1,4 glucose. So because we have a beta glycosidic linkage, the beta linkage is equatorial. And because the beta linkage is equatorial, we don't have a helical conformation like you do with starch. Instead of a helical conformation, you end up with straight chains. So cellulose actually does look like a bunch of hexagons linked together like so. And because we have a straight chain, we can have multiple chains lie side by side in the crystal, and they hydrogen bond with one another. So in pure cellulose, you end up with chains of cellulose organized side by side. They're actually anti-parallel, which is how I'm trying to draw it here. Not doing a very good job. Whatever. And the chains hydrogen bond with each other because we have all of these OH groups sticking off of the sides, and so these are free for hydrogen bonding with other molecules. So we end up with this very strong hydrogen bond network in cellulose. The hydrogen bond network in cellulose is so strong that it's actually insoluble in water, which is pretty remarkable because if you look at a molecule of glucose, that's a pretty polar molecule. You would expect that to dissolve in water. But in fact, the hydrogen bonding between the cellulose molecules is so strong that splitting it up to form new hydrogen bonds with water is energetically unfavorable. So it just doesn't dissolve in water. So cellulose is what the cell walls of plants are made of mostly, and also algae and some other organisms. Animals do not make cellulose, and only a few animals can break down cellulose as an energy source. Humans are not among them, so for us, cellulose is not useful as an energy source. It's just fiber. Chitin is very similar to cellulose, except instead of polymer of beta 1,4 glucose, it's beta 1,4 in acetylglucosamine. So chitin is like cellulose, but with a long non-hydrogen bonding branch hanging off of carbon 2. So this is that long non-hydrogen bonding branch. I guess technically it can hydrogen bond through the NH there. But we have an N-acetyl group on carbon 2. Otherwise, it's exactly like cellulose. So chitin is not as rigid as cellulose. And the reason for that is because this big, long N-acetyl group, this interferes with the packing between molecules, and therefore chitin is less rigid than cellulose. So chitin is found in insect exoskeletons and also shellfish exoskeletons, and it's also found in the cell walls of fungi. And then the last class of structural polysaccharides we're going to talk about are the glycosaminoglycans, or GAGs. These are the gelatinous ones. All right, so what is a glycosaminoglycan? The definition is so dry and so technical, I don't care that you know it. here's the features of glycosaminoglycans that you do need to know. Glycosaminoglycans are mostly in animals, although some non-animal creatures also make similar looking molecules. They are highly negatively charged, and the reason they are highly negatively charged is because they contain many carboxylic acid or sulfate groups, or I think in some rare cases nitrate groups as well. Okay? They tend to soak up large amounts of water because of that negative charge. And what they primarily do is they fill in the spaces between cells. And beyond that, they're diverse. So for example, hyaluronan, which is found in connective tissues. So what is that? So that's a gluconic acid, which is like glucose, but carbon 6 is changed to a carboxylic acid. Beta 1 to 3 in acetylglucosamine, beta 1 to 4, and then it repeats, right? So we have 2 monosaccharide repeats. There are others out there. So keratin sulfate is just plain old galactose, beta 1 to 4, in acetylglucosamine 6 sulfate, beta 1 to 3, and then it repeats again. Heparin is found inside the lining of blood vessels. It's got this really complicated structure where you've got both carboxylic acids and sulfates all over the place. So it's a strongly negatively charged molecule. What you should notice is that the space-filling diagram of heparin down here shows what the conformation of these things looks like in solution. Because of this super powerful negative charge, these molecules adopt extended conformations. So they don't fold up into well-defined shapes. Where you find glycosaminoglycans is on the surface of cells. Glycosaminoglycans extend outward from the plasma membrane of a cell, and they've got negative charges out the proverbial wazoo, and because of all those negative charges, they can form strong ion dipole interactions with water, and what that does is it soaks up water, which provides sort of cushioning for the cells. And that's important in higher animals because unlike plants, we don't have cell walls. So if it wasn't for these molecules, all our plasma membranes would be touching each other, and that would be a bad thing because the hydrophobic effect could cause all our cells to fuse, and then in a couple of minutes you would just be a single cell puddle lying on the ground. All right, so to summarize, GAGs have disaccharides repeats, lots of negatively charged groups, extended chains. Their function is to soak up water and to bind to proteins. And that's a function that is not entirely limited to glycosaminoglycans, but it's true for many other carbohydrates as well. So there is a term called glycoproteins, and these are just proteins with carbohydrates chemically attached. And usually they attach through glycosidic linkages to either serine, threonine, or asparagine. And we're going to leave it at that. And so glycoproteins are very important for molecular recognition, especially between different types of cells. So for example, certain cellular toxins, for example, cholera toxin, gets taken into cells because it binds to a certain carbohydrate on a certain molecule on the plasma membrane of cells. Glycoproteins are also involved in invasion of cells by pathogens. So, for example, viruses and some bacteria express proteins that bind to specific carbohydrates. And that binding is then responsible for those molecules either being fought off by immune cells, which would tend to also express certain carbohydrates on the surface so the cell knows not to destroy them, or for being taken into the cells, which is what we're showing here, and then targeted to organelles such as the lysosome. So I'm not going to talk about the function of glycoproteins in any detail, but if you're interested, you can see the discussion in that chapter of the textbook. Carbohydrate chemistry is important. It's not my favorite subject, but it is important. However, for the purposes of this class, we're not going to talk about this stuff. Our next subject is going to be the most important use of carbohydrates in organisms, and that is going to be carbohydrates as a fuel source, and that will lead us into metabolism. So in the meantime, I encourage you to do the carbohydrate worksheet. That has all the problems in as much depth as I care to ask you regarding carbohydrates. And then in the next lecture, we're going to introduce the unit on biological energetics. I will see you guys next time.